Life-changing delivery, how AAV works. It's not every day the future of science is discussed over pizza, but most Fridays worked for a group of young UF College of Medicine researchers in the late 1970s. They would talk about bacteria over pepperoni and viruses over a beer. They had some great discussions. Let's explore one discussion that's been going on for more than 30 years: gene therapy and AAV. Gene therapy means putting a new gene into a patient's organs to cure disease. To get this gene into the organ, you need a vehicle. The researchers modified AAV at a molecular level to create the perfect vector. Now you're probably asking yourself, what is AAV? AAV stands for adeno-associated virus, or as the researchers jokingly call it, an amazing virus, because it's the only known human virus that doesn't cause disease in humans. This makes it perfect for gene therapy. And AAV's single-strand genetic material is perfect to edit. Researchers use simple splicing techniques to remove the inside portion of AAV that they don't need and leave the ends that are needed for gene delivery. They then replace the inside portion with a new gene. As a result, they now have an AAV vector or vehicle to transport a new gene into the human body that will ultimately help the patient. They had to figure out how to make it. To clone the vector, they needed another virus to grow the AAV virus. A helper virus is used to help unlock the potential of AAV. They combined AAV with a bacterium, then they combined the helper virus with a bacterium to create something called a plasmid. Once they had the helper virus plasmid ready to go, the researchers introduced both AAV and the helper virus plasmids to a mammalian cell for replication. After introducing those two elements, they essentially had a factory for producing more AAV vector. They basically developed a molecular FedEx truck. Well, the next challenge was getting it where it needed to go. When the AAV vector goes into the human body, there are things on the surface of the virus that bind to things on the surface of the different cells called receptors. Researchers are trying to figure out how to modify that surface so that the AAV vector goes to exactly the right location or address. The corrective vector can reach its location via direct injection into the targeted tissue or by traveling through the bloodstream to its destination. Once the vector enters the patient, the only place it travels is to the heart, or the pancreas, or the brain, or wherever it needs to go to effectively help the patient. Upon arrival, our molecular FedEx truck begins producing or continually unloading the proteins necessary for that particular gene therapy. Basically, at the molecular level, we're delivering the payload, and the gene starts producing the desired protein. Using the AAV vector in human clinical trials, this technique has cured blindness, a type of genetic disease where the patient is essentially blind as the eye doesn't produce a certain protein. The AAV vector, with its corrective gene, is injected into the eye, and after the vector begins to make the protein, the patient can see. Hemophilia, a blood clotting disease. After the liver picks up the AAV vector, it produces proteins which save the patient from having to visit a clinic once a week for factor IX infusions. Heart disease. The AAV vector has improved the contractibility of the heart for people who are having arrhythmias. Now, after learning how to clone and utilize the AAV vector, we have a new tool that can be used to treat all kinds of diseases, especially genetic diseases. And that's how coming up with a self-replicating delivery system changed medicine forever.